Good morning, June.
Hi, good morning.
Can you hear me, June?
Yes, I can hear you.
Let me see. I think I have, am I mute or what? Yeah, I'm not mute. Okay. Good morning, June.
Yes. Do you
How are
hear
you, sir?
Yes, I can hear you. How are you?
Yeah. All right. You all set, ready to go. That's fantastic. Let's see. We have Dr. Finkel, Dr. Tagmeyer as our panelist. And do we have Lori on board and Asa? Let's see. Lori and I see. Let's see, where are they? Are they on? Let's see. I don't see them. All right. Let me see where they are. All right. I did email both of them last night. Did not let me. Oh, Lori has some. I'm here. I cannot speak. He says, team, would you activate Lori's microphone? And how about Asa? Tori, Lori, can you check your microphone? Alan, would you activate Lori's microphone? All right, let's see. Seems that Lori has problem with the microphone. Let me send him a message.
I'm here. The video won't activate.
All right, Lori's on. Lori on. That's great. So we're missing only Dr. Gustafson. I don't see her.
Yeah, the problem, AJ, it says I cannot start my video because the host has stopped it.
All right, very good, very good. So anyway, let me say a couple of introductory comments. Let us start the seminar. Asa will join us anytime she could. First, I would like to welcome everyone to our monthly webinar series by the Journal of Cardiovascular Aging. And today, we have a very special seminar, one of the most prominent and distinguished scientists of our time, Dr. Junichi Sadashima. Before we introduce Dr. Sadashima, I will tell you briefly about the journal. Our journal is an open access journal, and it has been around now for about 2 years. We publish quarterly, and currently there's no publication fee for publishing in the JCA. And therefore, we encourage everyone to consider submitting to JCA before publisher ultimately start placing publication fee for papers. It is, I think it's doing reasonably well, the journal. And let me put this on for you for a second. I don't know whether you can see my screen or not. We are publishing original research articles and review articles. We have other platforms as well. And we are cited a few citation indices, and we're hoping to get to PubMed very soon. We have a distinguished panelist of editorial board members. And really our approach is really empowering the authors rather than the reviewers or the editors. And if you have solid data, we're happy to work with you and get your paper published. I think if as long as data is valid and robust, we will be happy to have it reviewed very timely and fairly and get it published quickly for you. And we had a number of webinars. Here is the list of webinars you can appreciate. Today, webinars are about distinguished Sadoshima, named after Yoshinori Ohsumi, who won the Nobel Prize for his work on autophagy. And with that, our hosts are Asa Gustafsson and Lori Kirschenbaum. Princess Asa seems to be not here, but Prince Lori Kirschenbaum is here. And Lori, would you like to say a few words about Dr. Sadoshima, which is, we have biographed him here briefly and also posted his biography in our webpage and the announcement.
Sure, good morning. I don't know if you want me to give a little bit of a brief introduction for Dr. Sadoshima, very close colleague and friend. Just to give you a little bit of an overview of June's accomplishments and his background, because this would take probably about the seminar. For those of you who don't know Dr. Sadoshima, he received his medical degree in 1993 from Kyushu University in Fukuoka, Japan, and then went over to join the lab of Seigo Izumo from 1990 to 1994, and that's where he and I became quite acquainted as scientific trainees and colleagues over the many years. So June really was one of the key drivers of the early days of understanding mechanisms of cardiac hypertrophy and cardiac growth. And I still remember one of his seminal lectures on the role of angiotensin signaling and how stretch-induced pathways lead to cardiac hypertrophy. And I think after moving from Seigo's lab, he established his own lab at Rutgers and really was one of the key players in the world of autophagic signaling and cell death in the heart and the cardiovascular system. I don't know if really if people who don't know cardiovascular autophagy or cardiovascular signaling can actually pick up a research article or a review article with not coming across one of Dr. Sadoshima's many contributions that appear in really high impact journals such as Nature Communication, Cardiovascular Research, Journal of Clinical Investigation. I mentioned Nature Communication, Nature Medicine, some really seminal works that have really served as a platform for the field. And I think it's really testimony to his work that I just highlight some of the early prizes that he won. I'm really impressed, in fact, and this goes well beyond his demonstrating his ability of winning the Katz Prize in 1995, and then moving on to receiving the Janis Pfeffer Distinguished Lecturer Award from ISHR and Distinguished Investigator Award from BCDS from AHA. So his career has been reflected by not only his science excellence, but also by the acknowledgement and by our societies for his achievements. And that's really reflected also in the high number of publications and citations, well over 400 articles published. So I think with that, I'll stop and I'll turn it over to June. June, you're a dear friend and colleague. I'm delighted you're here this morning to give us this webinar on autophagy and this wonderful named lecture. So thank you again for the opportunity to introduce you.
Thank you, Laurie.
Okay. Do you see my slide?
Yes.
Okay. Great. First of all, thank you everyone for joining this seminar. And also I like to thank AJ and Rory for kind introduction. And it's been a pleasure working with Dr. Marian for the journal. And I hope this journal is growing. And today I would like to discuss the role of autophagy in aging. But unfortunately, my data regarding aging is not many. So I would say my presentation includes many preliminary results. So I would say work in progress. So autophagy is a very important mechanism for cellular degradation by lysosome and characterized by presence of autophagosome. And autophagy is completed by many involvement of many signaling mechanisms. For example, initiation of autophagy includes ULK1 and class 3 PI3 kinase, and also ubiquitin-like conjugation system, and also fusion of the autophagosome to lysosome requires small GTPase and snare system. And eventually content is degraded in lysosome and the content is recycled. And we started study of autophagy almost 20 years ago in collaboration with Dr. Stephen Wagner. He has a peak hibernation model, and we did unbiased screening using omics, and surprisingly top hit was all lysosome and autophagy related proteins and genes. And we found that autophagy exists in the area of hibernation, and in the area which has many autophagosome has less cell death, even in the presence of severe myocardial ischemia. So we predict that autophagy most likely supplying ATP and also involved in protein and organelle quality control mechanism to make the myocardium alive. And this study make me extremely excited. And we subsequently studied role of autophagy in the heart. Keep progressing, sorry. And to make the long story short, autophagy is activated by many cardiac stress. And in many cases, it does many good things. But unfortunately, in my opinion, the level of autophagy is not enough so that eventually cardiac injury and heart failure develops. And one of the mechanisms which makes autophagy insufficient is the presence of negative regulators in the heart. And one of our research focus is to find out the nature of negative regulators. And one of the most important one we discovered is MST1. It's a component of the Hippo pathway. And we found that the MST1 not only phosphorylate BCL-XL to induce apoptosis, but also phosphorylate BECRIN1. And this actually phosphorylation allows BECRIN1 and BCL2 interaction. And the BCL2 is a very potent inhibitor of BECRIN1, so that the activation of MST1 not only activate apoptosis, but also potently inhibit autophagy, and both of them induces cardiac dysfunction. And we show that, sorry, something is wrong in my computer. The MST1 is activated in human failing heart obtained from transplanted heart, extracted heart. And at the same time, BECRIN1, threonine 108 is phosphorylated and autophagy is inhibited. And to show the mechanistic role of MST1, we created permanent ligation in mice, dominant negative MST1 mice, normalized the survival, markedly improved the survival rate of mice after MI. But this survival advantage is disappeared if we cross these mice with loss of autophagy function mice. So we believe that MST1 activation is one of the mechanisms in which heart cannot activate enough autophagy. But the story is not so simple, and as like many biological functions in cells, autophagy can be dysregulated. And we believe that excessive autophagy activation is not good. And one example is autophagy activation during ischemia reperfusion. And particularly, we found that late phase of reperfusion, 6 to 24 hours, autophagosome vacuole is markedly accumulates in cardiomyocyte. And the 2014, Dr. Bess Levine's group, who already passed away a few years ago, but discovered autophagy, excessive autophagy can induce a special form of cell death. We call it autosis. And autosis is characterized by presence of various vacuole and also perinuclear space. We don't know why this perinuclear space shows up, but we find that perinuclear space can be observed 6 to 24 hours after reperfusion. And we believe that this accumulation of autophagosome is toxic to the heart because autophagosome formation uses endomembrane system so that the excessive formation of autophagosome can induce organelle dysfunction, mitochondria and ER function is decreased. And one of the important features of autosis is its sensitivity to cardiac glycoside. So according to Bess Levine, cardiac glycoside can inhibit autosis. Unfortunately, mice heart is not very sensitive to cardiac glycoside. So we got the humanized mice from Bess Levine. And then we gave wobine during the ischemia reperfusion. And we saw this perinuclear space is markedly reduced. And infarction size is also rescued. So although common sense in the field is the cell death occurs within few hours after reperfusion, but we believe that the persistent slow cell death due to autosis can occur, so that this can be the target of treatment. So the autophagy is very important during pathology of the heart, but sometimes autophagy is insufficient and sometimes autophagy is dysregulated, so that we really have to identify, evaluate the extent of autophagy during pathology of the heart. And if you think that the autophagy is insufficient, treatment to supplement autophagy is most likely efficient. But in case of dysregulation, suppression of autophagy is required. Some relatively non-specific drug to modify the level of autophagy is available. Although these are not necessarily specific, but I think if you use these reagents wisely, we may be able to treat the cardiac condition. So what's the role of autophagy in aging? So this is a very important question, and particularly I'm getting old and losing hair, so I'm very curious solving the mechanism. And according to the literature, the autophagy is generally decreases by aging. Many reports says autophagy declines. And several systemic gain of function mice exist. ATG5 Korean group and Becklin-1 Best-Levin's group. And we also have unpublished mice. And these mice show lifespan extension. The systemic autophagy activation is most likely good. And also many lifespan extension model, many organisms requires autophagy. So lifespan extension by caloric restriction requires autophagy. Without autophagy, lifespan extension cannot be observed. So we had a very good collaboration. We enjoyed, it's already finished, but we had the Duke Foundation with Guido Croma and Best-Levin and Rick Kitches, others. And we, in collaboration, we participate in Guido Croma's aging work. And we found that autophagic flux in aging mice is almost suppressed. So LC3 cannot accumulate even in the presence of roipeptin. So that I believe that aging cardiomyocyte cannot generate autophagosome, but it has a capacity to make it because spermidine can activate autophagy. And also Best-Levin's group made Becklin mutant. And this phenylalanine mutation actually prevent BCL2 binding so that the Becklin-1 is persistently activated. And both male and female mice show lifespan extension. And in her Nature paper, she described that aging cardiomyopathy is also prevented in these mice. So I believe that autophagy is involved in cardiac aging as well. So today I'm gonna show you my unpublished result. And I believe when you think about aging, we have to think of cell autonomous effect as well as autocrine, paracrine effects. And in terms of cell autonomous effects, I think we can easily speculate activation of autophagy basically improves cellular quality control mechanisms. But it's hard to figure out whether autophagy affects autocrine, paracrine, circulating factors or senescence, So I would like to address this issue next 20 minutes or so. So our hypothesis is suppression of autophagy directly stimulates senescence. And senescence occurs after proliferation in cancer in order to stop the presence of induction DNA damage and mutations. Cancer cells go into senescence. And whether or not cardiomyocyte undergoes senescence is a big question, but so far many people believe that the telomere dysfunction DNA damage induces senescence in cardiomyocyte. very similar to non-differentiated cells that include the cell cycle arrest markers, and also DNA damage markers, and also production of factors. We call it SASP, senescence associated secretory phenotype. And also the senescent cells can survive even in the presence of stress. So induction of anti-apoptotic mechanism is also important. So we examined whether autophagy suppression can induce senescence in cardiomyocyte. So we used ATG7 knockout mice. And this is very cool because cardiac specific knockout completely eliminate LC32, and P62 markedly accumulates. So this is very useful mice studying the role of a conventional autophagy. And in these mouse heart, we saw markers of senescence are increased. And this is IL-6 and CTNT staining, and we confirmed that the IL-6 activation is observed in myocyte. And also ATG7 knockout mice develop cardiac dysfunction. So you may say that senescence is secondary to cardiac dysfunction. So we injected AAV-CRE so that only few myocyte deplete ATG7, chimeric mice, and cardiac function is obviously normal. And we still see the green cells have more the DNA damage markers, gamma H2AX. And also we can induce senescence markers in cultured cardiomyocyte. So we believe that the suppression of autophagy has the ability to induce senescence in cardiomyocyte. So, but since in the heart, adult heart, cardiomyocyte is essentially non-dividing, maybe divide very slowly. So question is whether we should get rid of senescent cells. As you know, the senescent cell can produce factors inducing inflammation and senescence in surrounding cells. But myocyte is a myocyte. So we have to weigh which is more critical, preserving contraction or inhibiting SASP. So we used 6 to 7 months old ATG7 knockout mice, which already show sign of cardiac dysfunction and senescence. And then we treated mice with navitoclax, it's a BCL2 inhibitors. And we found that this gamma H2AX cells decreases after ABT treatment, and function is improved. So whether senescence is irreversible is things of controversial area, I believe, but I think this result suggests that the killing senescent cell may be good. And we also have a doxorubicin senescence mouse model. And Joe Hill's group, we also confirmed this observation, but since he reported this much earlier than our group. So he looked at effect of doxorubicin upon autophagic flux, and he found that autophagic flux is inhibited in the presence of autophagy. The bufferomycin cannot increase LC3/2. So we believe that the doxorubicin model is another model in which autophagy is inhibited and senescence is induced. And we crossed our own gain of function autophagy mouse model, Best11 mutated phenylalanine, but we mutated threonine 106. This corresponds to 108 in humans. But in these mice, autophagy is higher. And we are able to show gamma H2AX positive cell is decreased. So, and also our function is improved. So we believe that the suppression of autophagy induces senescence, and senescence may induce the cardiac dysfunction. And sorry, this is the effect of nabitoclax upon doxorubicin cardiomyopathy. So in this experiment, we intentionally gave ABT after 3 doses of doxorubicin. So by the time when we give nabitoclax, the senescence is already increased. And then we measured number of senescent DNA damaged cells, and we found that nabitoclax can decrease senescent cells and improve cardiac function. So in 2 models, autophagy ATG7 knockout mice and doxorubicin knockout mice, autophagy suppression induces senescence, and senescence may induce cardiac dysfunction. So then we wanted to know what's the underlying mechanism. Now, of course, there are many mechanisms inducing senescence. But a few years ago, we published a paper. This has nothing to do with aging, but we studied the lysosomal storage disease, and in which lysosome function is markedly inhibited. And surprisingly, in those mice, YAP accumulates, and it turned out that the YAP interact with P62. This is the ubiquitin binding and also LC3 binding adapter protein. And the YAP P62 binding induces YAP degradation through autophagy. And if you stop autophagy, YAP accumulates. So actually, in fact, that we found that YAP accumulates in ATG7 knockout mice. And also in the presence of DNA damage, YAP go into nucleus. This is a staining of YAP, and the doxorubicin treatment. This is a control. Stupid mistake, but the doxorubicin induces nuclear translocation of YAP, and this actually translocation was inhibited in the presence of ATM inhibitor. And this translocation is also observed in doxorubicin treated heart, and CTGF is upregulated. So this is relatively faint, but this is due to the fact that CTGF is upregulated only in YAP activated cell, not every cells. So we believe that in case of at least the doxorubicin, two mechanisms can activate YAP. So we immediately decide to make cross mice. So we crossed the ATG7 knockout and YAP knockout mice, and we are able to show that the senescence is decreased and function is improved. But before we plan this experiment, we had a concern that since we previously reported YAP is a cell survival mechanism, and in, for example, acute phase of pressure overload and other conditions, YAP is protective. So generating, crossing two mice, both are potentially harmful, may not be good. So before we planned this experiment, we prepared alternative strategy. And we found an interesting paper in Cell Metabolism in which P16 high promoter is placed before CRE so that the CRE can be expressed in P16 high cells. Many of them are senescent. So we copy the design from Cell Metabolism. We can quickly synthesize DNA and send it to genome editing core, and then made a P16 high CRE mice. And we crossed those mice with the TD tomato, rosa TD tomato cells, mice. And we treated cross mice with doxorubicin. And the doxorubicin increased TD tomato positive myocyte. And also we found that those myocytes are relatively bigger than the TD tomato minus cells. And also recent results show that these mice have higher IL-6 positivity. So we basically crossed this P16 CRE mice with YAP frox mice. And the idea is to knock out YAP in P16 high cells. And we found that these mice, in the presence of doxorubicin, we have a persistent version and also the inducible CRE-ERT version. But in this case, tamoxifen plus doxorubicin treatment decreased the YAP positive cells and also senescent cells, as you see here. And very interestingly, if we eliminate YAP only in a senescent cardiomyocyte, can improve ejection fraction and decreases cardiac fibrosis. So we are thinking that YAP is doing something in P16 high cells, most likely senescent cells. So we don't know the mechanism yet, but the one possibility is that YAP promotes secretion of some factors. And this has been shown by many prominent investigators. And Jim Martin reported in cardiomyocyte, YAP can promote secretion of Wnt ligands. And also I think this is Eric Olson's group, macrophage. YAP can increase his IL-6. And Dominic Derry department also reported YAP can increase IL-6 secretion from cardiac fibroblasts. And John Epstein's group reported epicardial cells can secrete something by YAP. So one mechanism, possible mechanism, is YAP can produce some factors. And we are actively investigating this mechanism right now. And another important thing in SASP is maintenance of energy. As you know, the protein translation requires ATP. So if you want to actively secrete factors, those cells must be very healthy. And one report suggests that mitochondria function need to be maintained. And I really wanted to know whether this is the case. It's interesting that young non-senescent cells requires autophagy and mitophagy, mitochondrial quality control mechanism for survival. But the senescent cells also requires quality control mechanism. And this must be conducted even in harsh environment. So, but obviously quality control mechanism is good for young normal cells. But in terms of aging perspective, this actually quality control may not be necessarily good. But we are so curious how then this happens. So we treated cardiomyocyte with doxorubicin, and then examined autophagy flux. And as Joe Hill reported, we saw autophagic flux is inhibited by doxorubicin. But very interestingly, mitophagy evaluated by mitochema is activated. And there's clear dissociation between ordinary autophagy flux and mitophagy. And we recently reported that this happens in the heart in the presence of stress. For example, pressure overload, ischemia, and diabetes. In many chronic cardiac conditions, general autophagy run out, but the mitophagy is not. So we examined whether this is true in another model, and we published the paper in Journal of Cardiovascular Aging in 2021. Pei-Yon Zhai made a ULK1 knock-in mice in which autophagy is suppressed. And the GSK3 beta phosphorylate ULK1 and promote autophagy. But if you mutate this phosphorylation site, these mice have decreased autophagic flux. And P62 accumulates. And we found that in these mice, mitophagy is still activated. So I think that this is a key for senescent mice, senescent cells to survive even in a harsh environment. So they activate BCL2 and also upregulate mitochondrial quality control mechanism. So then how then this dissociation between autophagy and mitophagy occurs? In our hypothesis is that the heart has alternative mechanism. Mitochondrial quality control is such an important mechanism. So they have a way to degrade mitochondria, even in the presence of complete downregulation of a general autophagy, and we reported that such mechanism to induce mitophagy exists in a heart. In the lucky, we have a loss of function mouse model in which alternative mitophagy is inhibited while alternative or conventional mitophagy is maintained. And in these mice, we gave doxorubicin, and we are able to show increases in senescence, and development of cardiac dysfunction was attenuated. And also, we recently made a cardiac-specific version of this loss of function alternative autophagy mouse model, and we observed the same things. And so involvement of YAP, we are not certain, but at least YAP can induce activation of a mitophagy. And also, we have previously shown that the YAP can interact with TFE3. It's a master regulator of a lysosome and autophagy gene expression. So we are thinking that although why YAP induces senescence and SASP, we don't know yet, but we speculate YAP in senescent cell may induce SASP, and mitochondrial quality control mechanism. So to summarize my talk, although this is still at the stage of hypothesis, I would say, but suppression of autophagy accumulates a YAP. And accumulation maybe is enough to induce increases in nuclear YAP. But if there's a stress, DNA damage or something, it can induce nuclear localization of YAP. And once YAP is activated in senescent cells, it not only promote survival, also try to preserve mitochondrial function and SASP, and induces the autocrine paracrine mechanism of aging. So when you think about aging, we have to think how to prevent aging. But if our hypothesis is true, we should also think how to treat aging. And apparently these are very different approach. One is to promote survival of cardiomyocyte, but the other is to kill cardiomyocyte. And still, I don't know whether killing cardiomyocyte is a good thing, but if this hypothesis is true, like a cancer, cancer, we have to preserve normal cell, but we have to kill cancer cells. So we have to distinguish what is a unique feature of senescent cells, and think about this, what makes senescence unique and bad. Activation of senescence pathway and SASP mechanism, preservation of energy is very important. So I think these feature, it could be the target of treatment in senescent cells. And I propose that these could be potentially selectively cured. Since these mechanisms are not so strongly activated ordinary cells, so even preventing this may not affect much in ordinary cells. So I think, we are looking for a specific mechanism in cancer cells for cancer treatment. We believe that this could be the approach, and I propose some mechanism allow us to cure only senescent cardiomyocyte. So this is the final slide. Although I focus on autocrine paracrine mechanism and treating senescence, but I believe that preventing senescence is also very important. And I believe that autophagy, in this case, autophagy, mitophagy activation is a good thing. And this is my favorite paper published a few years ago about intermittent time-restricted feeding paper. Although this is a Drosophila paper, but the 20 hours of fasting every other day, targeting night. The autophagy gene is regulated by circadian, Laurie's favorite subject. But adding senescence, adding the fasting at the same time during the night dramatically facilitate autophagy, and this prevent aging and promote longevity. And the restricting food is terrible. I just can't do it. So, but probably intermittent fasting is the way to go. I'm not sure whether this is very healthy. Some paper, recent cell metabolism show that intermittent fasting is not always good in doxorubicin cardiomyopathy, but once in a while you clean up your heart is most likely good. So like a car wash, I wash my car 3 to 4 months. So once in a while you clean your heart is good, and this is probably a way to prevent our aging. So overall, I wanted to tell you that autophagy is most likely involved in aging in the heart, and identifying the timing to stimulate autophagy or inhibiting autophagy is a key, and possibly we are able to modulate the process of aging. So finally, I'd like to introduce my fellows, and most of the work I presented today was done by Pei-Yong, who is a senior fellow, and also Una-san, she's a student of my lab. Thank you for your attention.
Thank you, Jun. It's fascinating how biology is complex. It's amazing what's going on inside the cell. With that, I will ask the first question, if Laurie permits me, and then Dr. Tagmeyer and Dr. Torren Finkel are the panelists to guide the discussion. My question relates really ATM targeting YAP. And if ATM directly targets YAP and phosphorylates it, so it inactivates YAP, right? And that inactivation will lead to less YAP in the nucleus, and therefore suppression of YAP activity, while the effect seems to be YAP also involved in mitophagy when activated. I'm a little bit conflicted how to resolve this observations.
So if you look at the serine 127, the site that many people investigate, the aging and doxorubicin decreases 127. So that consistent with YAP activation. And also in terms of DNA damage pathway, tyrosine 357, I guess, phosphorylation actually activates YAP. And we are not sure probably ATM activates other tyrosine kinase to induce phosphorylation of tyrosine 370, 57. And we are actively looking at this possibility. So how phosphorylation is regulated is not completely understood, but we believe that YAP comes to the nucleus and also activates downstream targets such as CTGF. So I believe that YAP is activated somehow.
Thank you, June. Dr. Teigmeyer, Dr. Finkel, Lori, I'll leave it up to you all to guide the conversation.
There was a question, June. First off, thanks very much. It was a really brilliant overview of the autophagic process and how it can tie to senescence and aging. I had one quick question, and then there was a question that we have maybe from the audience, and then we'll switch it over to Torun and Heinrich. I was really curious that proteasome inhibition. I mean, maybe you showed it, but I didn't see it. Proteasome inhibition had no effect on YAP turnover. It was basically mediated by autophagy. Is that did I interpret that right? Is YAP turned over by the proteasome?
YAP. So we believe that the YAP is degraded by autophagy, so not the proteasome.
Okay, so it makes sense that it would accumulate if autophagy is impaired. There was a question from.
So P62 is the sorry, I'm get confused. Just forget.
Okay, that's fine. We can chat another time on it. There's a question from Priya Gura, who asks, how does autophagy directly regulate DNA damage? Is it dependent upon YAP?
So DNA damage pathway is sometimes protective, right? In terms of the induces survival even in the presence of DNA damage. But I think that actually induces activation of a P53, and P53 possibly is involved in SASP. So how it does many probably harmful thing is not yet solved, but I believe that there could be a mechanism through P53.
Excellent. Okay, I'll head it over to Torun and to Heinrich.
Thanks, Lloyd. That was a great talk, June. Good to see you. I guess one of the questions I had is you talked a lot and very nicely and comprehensively about the cardiac myocyte, but what about the other cells in the heart and these mechanisms? What role do you think endothelial or fibroblast or immune cell senescence is playing in aging or in these various models of stress-induced cardiac dysfunction, and maybe what the crosstalk between cardiac myocyte senescence and other cell type senescence in the heart?
Yeah, Torun, that's $1 million question, and I do not exclude involvement of other cell types. And in fact, that the Una-san, my student, is working on Wnt5a in doxorubicin. But this, very interestingly, Wnt5a expression is mostly in cardiomyocyte. So in some condition, cardiomyocyte is involved, but in order to elucidate the role of other cell types, cell communication, we really have to do single cell RNA sequencing and other mechanisms, other interventions. So that's the next step.
Great. Heinrich, did you want to ask questions? I don't know the format, if we alternate or if I ask all my questions.
Whatever, it's open, whatever Professor Marian allows us to do. But I think Heinrich, you're up. If you have any questions for June.
Thank you. Can you hear me all right?
Yes.
June, this was fantastic. I liked every slide you showed. Some of them perhaps even more than others, but fantastic. And your finishing note on the time-restricted feeding, I think, is very appropriate because this is a largely unexplored area of cardiovascular health and metabolism. And I have one question that relates to the overall theme of myocardial protein turnover that I've been involved in since my early days with Mike Lesh 50 years ago. You mentioned that suppression of autophagy promotes doxorubicin-induced cardiomyopathy. Have you or anyone that you know of ever actually addressed a myocardial protein turnover with a straightforward post-chase approach that that would give an insight into dynamics of the internal self-renewal of the cardiomyocyte? That's my question, the protein turnover.
So, Dr. Tegmeyer, I think your question is very important, and we saw autophagy flux is suppressed, and probably it's important to look at the protein turnover in my system, but we haven't done it yet. So that's also an important question to be addressed in the future.
Yeah, a comment on the side of your YAP story that just reeks of metabolism. I'm sure you will make many important discoveries in the future.
Lori, one quick question. June, does your CDKN2A CRE affect expression of the CDKN2A alone itself? Because it's inserted after exon 2. I wonder whether it shuts down CDKN2A, which is, of course, is a cell cycle regulator.
So that's a good question. How can I? So how can I measure it? How can I determine it? So you are asking a P16 high CRE, right?
Right. Is the expression of P16 is reduced?
Yeah, P16.
Because
I guess I was confused. I mean,
after exon 2, and I wondered that would affect the expression of this cell cycle regulator itself.
June, is that CRE expressed in the endogenous P16 locus, or is that a?
Right, it is not into endogenous P16 locus.
Or is that just a promoter?
It's an endogenous locus. So that's a good question. We haven't.
Another question I was wondering, it seems to be that a degree of improvement in LV function, which is practically normalized with your senolytic therapy, it seems to be disproportionate to number of H2AFX positive cells, which is 0.01% or 1 in 10,000 cell or so, or at most 1 in 1,000 cell. And so I wonder whether there are other effects beyond targeting the senolytic, cells, because targeting, 0.1% of the myocyte is not going to impact cardiac function. There might be alternative mechanism by which this senolytic therapy normalizes cardiac function, basically.
Yeah, so the effect may not be limited to senescent cells, because you said that the proportion number is too low. So I think we have to find out what kind of cells are affected, but that's also next step.
Thank you, Jun. Any other questions from the audience or from panelists? We missed Dr. Gustafsson today. It was unfortunate, but it's California time, 6 a.m. maybe, that was the reason. June, we're grateful to you for fantastic, really, it just shows the complexity of biology and multiplicity of the functions of each single protein involved in various mechanisms. It's just, it's amazing how complex biology is. With that, I'd like to acknowledge a few people on the panelists. I see Dr. Daniel Kleonsky is there, one of your colleagues, and we appreciate his participation. I see Dominic, one of your former trainees, is in the program, and I see some of my former colleagues. Dr. Lombardi is up there as well. My gratitude to all for participation. With that, again, my appreciation to Dr. Torren Finkel, Dr. Tagmeyer, and our distinguished host, Dr. Lori Kirschenbaum, who seems to be exposed too much to sun and seems to be a little plethoric. He may need to give some blood to reduce his hemoglobin level. And June, our fascination with your science and our sincere gratitude to you for all you do for the journal and your camaraderie as a colleague and a scientist, moving the field forward with very, really amazing effort and with fascinating discoveries. Our gratitude to you. With that, we will close today's webinar and appreciate everyone's participation. I'm looking forward to the next, and any of you who has a candidate that you wish to host, please let me know. We will make the arrangement. Thank you all very much, and you all have a fantastic day. Great. Thank you, June. Bye-bye.